My name is Christian Haass. I'm working at the University of Munich, and in parallel I'm the speaker of the German Center for Neurodegenerative Diseases. A long time ago, when I was a PhD student in Heidelberg, so the neighboring lab was working on Alzheimer's disease, and it was Konrad Bayreuther, who by that time cloned the APP gene, and who is now basically the father of modern AD research. And I collaborated with him quite a bit, not on Alzheimer's disease, but by that time on the proteasome system. And the enthusiasm of Konrad basically brought me into the field. My main contribution, I think, goes in two different parts. So the very first part was focusing on the generation of amyloid, which is the component which forms the amyloid plaques. So by that time I was working at the Harvard Medical School, and what we found by the time was that amyloid is produced normally by all of us, which indicates that we all have a very high risk to get the disease. And basically with that finding, we had a system in our hand in cell culture to identify the mechanisms how amyloid is being produced, how it's processed by which protease, I call it secretases, and importantly, we also had a system in hand to basically investigate how mutations which force Alzheimer's disease in families via genetics, how they are working. So we made a very surprising observation by the time that one of the mutations which is associated with familial Alzheimer's disease simply increases amyloid production. So the first part of my career was basically focusing on amyloid itself, on the amyloid cascade, on the proteases involved to generate amyloid. And the second part then of my career, which I started maybe about 14 years ago, is now on neuroinflammation, because there were risk factors identified which enhance the chance for Alzheimer's disease, and these risk factors are exclusively expressed in microglia cells. And that really also made me a bit worried, because I was a strong believer in the amyloid cascade, and the amyloid cascade is of course neuronal driven, and now microglia came into the game. So I wanted to prove myself basically, and so I thought, let's find out what these risk factors are doing. And there we made a super surprising observation. So by the time everybody believed that microglia are doing something bad for the pathology, so they drive the pathology in the AD brain, and we found exactly the opposite. So the mutations which were found in the microglia cells, they were loss of function mutations, and they stopped a protective function of microglia. So that was a complete unexpected result. At the same time, gave us completely new insights into the disease mechanisms and new targets for treatment. The question, of course, is how do microglia behave throughout disease progression? What we found, I think, plays the biggest role at the beginning of the disease. So the microglia early on are there as immune cells in the brain to defend the brain against foreign material, in that case amyloid plaques or developing amyloid plaques. But in the long run, of course, then the microglia can be overwhelmed and start also toxic processes later during the disease. I'm in the field of Alzheimer's research now almost on the days since 35 years. So I basically watched it from the very beginning of the molecular pathways until now. And if you would have asked me 35 years ago, do I ever see a medication which removes amyloid plaques and which may have at least some effect on cognition, I would have by that time said, no, impossible, this will not work. But there are antibodies now available against amyloid, and we all know that these antibodies rapidly remove amyloid plaques from the brain, and this has quite dramatic consequences because the entire amyloid cascade seems to be interrupted. So even the tau protein, which forms the tangles, is reduced when you reduce amyloid plaques, and there's at least some effect on cognition. So decline of cognition is slowed down at least a little bit. I think it's a very good beginning. There are major side effects which have to be solved, absolutely clear, there's no question about it. But at least I think this is a start point to develop better antibodies, better treatments, and I think most importantly to understand when to treat. I think that this is the most important thing what we need to do right now. I think the antibodies have a big potential if they are given early enough, basically before even the disease really starts. When the first amyloid seeds are precipitating, by that time the antibody should be already around. I'm relatively optimistic that we go into the right direction, but still we need another decade to develop antibodies or other drugs which really help cognition. A research prize is always something really great, because I think what people from outside don't understand, the way how we work is not like normal people work. We don't have an 8-hour working day. We work like 12 hours a day, 14 hours a day. Sometimes it's really frustrating. Experiments are not working. We get contradictory results and we don't know how to continue. So sometimes this is really a frustrating process, and you really need a lot of excitement to live through this progress. And for that, I think all these awards are absolutely fantastic, because they are stimulating scientists, and they show scientists some appreciation. And this prize specifically, if you look at the previous prize winners, I mean, it's the who is who in Alzheimer's disease research. And if you look at last year, when Lars Landfeld got the award, he is the person who developed the amyloid antibodies, which are now in the clinic.